Welcome to a fascinating journey into the frigid waters of abandoned mines near Piesalmi, Finland, where nature's tiniest organisms are leading a revolution in sustainable resource extraction. Scientists have deployed specialized bacteria capable of extracting gold from mining waste with remarkable efficiency, a breakthrough that could transform the global mining industry while dramatically reducing its environmental impact. Today, we'll explore how these microscopic miners are changing our relationship with Earth's resources and potentially solving some of our most pressing environmental challenges. The mining industry has traditionally relied on mechanical excavation and harsh chemical processing to extract precious metals from the Earth. But in Finland, researchers are pioneering a fundamentally different approach. By harnessing microorganisms that have evolved over billions of years to interact with mineral substrates, scientists at VTT Technical Research Center are essentially recruiting nature's own metallurgists to solve one of modern industry's most pressing challenges: obtaining critical resources without devastating ecological consequences. This isn't just a local Finnish innovation. It represents a potential solution to global resource scarcity, environmental degradation, and even the mounting electronic waste crisis that threatens ecosystems worldwide. At the heart of this revolutionary approach are two remarkable microorganisms: Acidithiobacillus ferrooxidans and Leptospirillum ferrooxidans. These are extremophile bacteria that thrive in highly acidic environments where most organisms would perish instantly. Dr. Maija Korhonen, who leads the research team, has harnessed their unique metabolic pathway that allows them to oxidize sulfide minerals surrounding gold particles. This process effectively dissolves the mineral matrix while leaving the precious metal intact. As Dr. Korhonen explains, these microbes evolved to extract energy from minerals in ways we're only beginning to understand. They're essentially performing microbial alchemy, but with solid scientific principles behind it. The 2023 pilot project demonstrated something remarkable: recovery rates of up to 83% from tailings previously considered depleted. These tailings contain gold concentrations as low as 0.5 parts per million, far below what conventional mining would consider profitable. The bacteria are literally turning waste into treasure. What makes these bacteria truly fascinating is their specialized cellular machinery. Their cell membranes contain unique metalloproteins that facilitate electron transfer from mineral surfaces. Additionally, they secrete specific organic compounds that act as chelating agents, binding to metal ions and facilitating their transport across cell membranes. This biological machinery wasn't designed in a lab. It's been refined through evolutionary processes occurring in extreme environments for millions of years. Nature has essentially created mining specialists far more sophisticated than any human-engineered solution. The bacterial communities also demonstrate remarkable adaptive intelligence. When introduced to new mineral substrates, they undergo rapid genetic expression changes, upregulating genes associated with specific metal processing pathways. Through horizontal gene transfer, beneficial adaptations spread quickly throughout the microbial population, creating a constantly evolving extraction system that optimizes itself for the specific mineralogical conditions present in each mining waste deposit. What makes this approach particularly valuable in Finland is its temperature adaptability. Unlike conventional bioleaching operations in warmer regions, the Finnish research team has identified cold-adapted strains that maintain metabolic activity at temperatures as low as 4 degrees Celsius. The bacteria we've isolated represent a distinct ecotype that has evolved in the Nordic mining environment, notes microbiologist Dr. Jukka Nieminen, co-author of the study. Their cold adaptation mechanisms involve modified membrane lipid structures that maintain fluidity at temperatures that would render tropical strains dormant. This cold tolerance extends the operational season for bioleaching in northern climates by approximately 4 to 5 months annually compared to conventional methods. These psychrophilic or cold-loving adaptations include specialized enzymes with lower activation energies, allowing biochemical reactions to proceed efficiently at temperatures that would significantly slow metabolic processes in mesophilic bacteria. The cell membranes contain higher proportions of unsaturated fatty acids, preventing the rigidification that typically occurs in cold environments. Additionally, these bacteria produce antifreeze proteins that inhibit ice crystal formation in their immediate microenvironment, allowing them to remain active even when the surrounding water approaches freezing temperatures. Climate modeling suggests this cold adaptation capability will become increasingly valuable as mining operations expand into previously inaccessible northern regions exposed by retreating permafrost. The bacteria effectively transform a regional climate limitation into a competitive advantage, potentially positioning Nordic countries as leaders in sustainable mining biotechnology. In an era of climate change, When traditional industries face disruption, this adaptive approach represents a forward-thinking solution that works with changing environmental conditions rather than against them. The environmental benefits of this bacterial mining approach are substantial. Traditional gold extraction typically relies on cyanide leaching, a process that generates toxic waste and requires extensive water usage. The bacterial approach eliminates the use of cyanide entirely while reducing water consumption by approximately 60%. From an economic perspective, the process creates value from existing waste. Finland alone has an estimated 100 million tons or more of legacy mining tailings, many containing trace amounts of precious metals that were uneconomical to extract using 20th century methods. Preliminary economic assessments suggest that bacterial extraction could yield up to 140 million euros in gold from just the Piosalmi tailings. This technology aligns perfectly with Finland's critical minerals strategy, addressing resource security concerns in an era of increasing geopolitical mineral competition, particularly with Russia, a former significant trading partner for raw materials that is now less accessible due to geopolitical tensions. Beyond the direct economic benefits, this approach creates an entirely new category of mining that fundamentally inverts traditional extraction economics. Conventional mining becomes progressively less economical as ore grades decrease, requiring more energy and generating more waste per unit of metal recovered. In contrast, bioleaching becomes more competitive precisely when dealing with low-grade materials that would otherwise be classified as waste. This inverted economic model creates financial incentives that align with environmental objectives, a rare alignment in resource extraction industries. The process also generates significantly lower greenhouse gas emissions compared to conventional mining. Life cycle assessments indicate a 72% reduction in carbon footprint per kilogram of gold recovered. This reduction comes primarily from reduced energy requirements for crushing and grinding, elimination of high-temperature smelting processes, and lower transportation needs since the processing occurs directly at the waste site. Perhaps most intriguing is the research team's next frontier, adapting these bacterial systems to extract gold and other precious metals from electronic waste. Laboratory tests have demonstrated that modified strains can effectively recover up to 92% of gold from crushed circuit boards. The bacterial genomes contain remarkable metabolic flexibility, explains Dr. Nieminen. We've identified gene clusters that can be upregulated to process different substrate materials, including the complex polymer metal matrices found in e-waste. This application could help address the growing global e-waste crisis, which generated 53.6 million metric tons in 2023 alone, with less than 20% currently recycled. The adaptation to e-waste processing required significant bioengineering innovations. Electronic components contain numerous synthetic compounds never encountered in natural environments, presenting novel challenges for microbial metabolism. Researchers have employed directed evolution techniques, exposing bacterial cultures to progressively higher concentrations of e-waste materials and selecting for strains that develop effective processing capabilities. They've also identified specific plasmids, mobile genetic elements, that confer resistance to toxic compounds found in electronic components, such as flame retardants and various polymer additives. Most remarkably, the bacterial communities demonstrate an unexpected ability to degrade certain persistent organic pollutants common in electronics while simultaneously extracting precious metals. This dual functionality transforms what would otherwise be a complex waste management problem into a valuable opportunity for resource recovery. The bacteria aren't just mining, they're actively cleaning up toxic compounds that would otherwise persist in the environment for decades. This unexpected benefit has opened up entirely new research directions in bioremediation, suggesting these specialized microbes might help address legacy pollution issues beyond just resource extraction. In an unexpected cross-disciplinary development, the research team has incorporated traditional Finnish mining knowledge into their biotechnology approach. Historical records from 18th century copper mines revealed that certain wooden support beams, when submerged in mine water, appeared to accelerate the precipitation of metal. What 18th century miners observed empirically was likely early biofilm formation on wooden surfaces creating microenvironments favorable for metal concentration, explains historical mining expert Dr. Liisa Mäkinen, who collaborated on the project. We've engineered modern biofilm support structures based on these historical observations. This integration of centuries-old mining practices with cutting-edge biotechnology exemplifies how traditional knowledge can inform modern scientific approaches. The wooden structures provide ideal surfaces for bacterial attachment and biofilm development, creating complex three-dimensional communities with specialized microniches. The cellulose and lignin components gradually degrade, releasing compounds that serve as additional carbon sources for heterotrophic bacteria that support the primary metal-extracting species. This symbiotic relationship enhances overall system efficiency while reducing the need for external nutrient additions. Archival research uncovered detailed journals from Finnish miners dating back to 1742, describing specific wood types and aging processes that appeared most effective for metal extraction. Modern analysis has revealed that these traditional preferences correspond precisely with wood characteristics that optimize bacterial attachment and biofilm formation. Knowledge developed through generations of empirical observation, now validated through microbiological research. The pine and spruce varieties specifically mentioned in historical accounts contain resin compounds that modern analysis shows create microenvironments particularly conducive to metal-binding bacterial communities. This remarkable convergence of traditional knowledge and cutting-edge science demonstrates how valuable historical practices can be, even in our most advanced technological endeavors. As the technology scales up in 2024 with a planned 10,000-ton demonstration facility, it may represent the beginning of a new chapter in resource extraction, one where microscopic organisms do the heavy lifting while humans minimize their environmental footprint. This isn't just a minor improvement to existing mining techniques. It's a fundamental reimagining of how we obtain resources from the earth. The demonstration facility will serve as both a commercial proof of concept and a research platform for further refinements of the process. Engineers have designed modular components that can be transported to remote mining waste sites, allowing for on-site processing that further reduces the environmental footprint of the operation. The principles behind this microbial mining extend beyond gold extraction. The same bacterial communities have demonstrated the ability to recover copper, zinc, and even rare earth elements, which are essential for renewable energy technologies. As global demand for these materials increases with the transition to green energy, bacterial extraction could provide a sustainable pathway to meet these needs. Researchers have identified specific bacterial strains that show affinity for different metal groups, allowing for targeted recovery operations. This versatility makes the technology applicable to virtually any mining waste site worldwide, regardless of the specific mineral composition. This biotechnological approach fundamentally reframes our relationship with Earth's mineral resources. Rather than viewing mining as an inherently destructive process of extraction, it positions resource recovery as a form of participation in natural biogeochemical cycles that have been occurring since life first emerged. By aligning human technological systems with biological processes that have evolved over billions of years, we potentially create more sustainable pathways for meeting material needs without compromising ecological integrity. As one Finnish mining engineer quipped during the project presentation, "For centuries, we've been digging into the earth with machines. Now we're letting nature's smallest organisms do the work with remarkable precision. Perhaps this is how mining was always meant to be." This microbial mining revolution represents not merely a technological advancement but a philosophical shift, recognizing that some of our most sophisticated solutions may come not from conquering nature but from becoming better students of its ancient expertise. As we've explored throughout this journey, the bacterial gold mining revolution in Finland represents far more than a technological innovation. It embodies a profound shift in humanity's relationship with resource extraction. By harnessing microorganisms that have evolved sophisticated mineral processing capabilities over billions of years, scientists have created a system that transforms mining waste into valuable resources while dramatically reducing environmental impact. From the specialized cellular machinery of extremophile bacteria to their remarkable cold adaptation in Nordic environments, from their application to electronic waste to the integration of traditional Finnish mining knowledge, this approach demonstrates how biological processes can outperform conventional industrial methods. As the 10,000-ton demonstration facility comes online in 2024, we stand at the threshold of a new era in mining, one where microscopic organisms become our partners in creating truly sustainable resource cycles. The philosophical implications are as valuable as the gold being recovered. Perhaps our most sophisticated technologies aren't those that conquer nature, but those that learn from and work within natural systems that have been perfecting their processes since life first emerged on Earth. In the frigid waters of abandoned Finnish mines, bacteria are teaching us that lesson one gold particle at a time. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.